Hello and welcome back, everybody, to our last talk of today's session. So, we'll be hearing very shortly from Dr. Eleanor Raffan, who will be telling us about the lessons from Labradors on how genes cause obesity. So, I will pass it off then to Eleanor.
Thanks, Dylan. So just to check, can everyone see you can see my screen and we're good to go. Lovely. Well, it feels so funny. Last time I did one of these Science Festival talks, we were in a proper big hall, and it's such a shame not to be able to have that same atmosphere today. But hopefully this format means that more people can reach this and see it over time. So it's very nice of those of you who've come along live, and hopefully a few more of you will watch later on. My name's Eleanor Raffan. I am a vet and lecturer at the Department of Physiology, Development and Neuroscience. My background was very much as a clinical vet, and then I kind of got the bug for research and science, and I've been very lucky for the last few years to be able to combine those two things in studying dogs, a little bit like Woody here. I love this little video that came from a friend of mine. It was her dog, and over his life, Woody got into all sorts of scrapes in the pursuit of food. I don't know how the timing on the videos is. A previous speaker said they were a little bit off, but you probably got that cute little whimper at the end. And he was an absolutely typical Labrador, really highly food motivated, always on the lookout for a scrap of food that would satisfy his food craving. And I think studying dogs like Woody is well worthwhile because as a vet, I can see the clinical importance of weight gain and obesity in dogs. But primarily, I'm also interested in contributing to the study of human obesity and our understanding of it. And that's really important because there is a global obesity epidemic, and obesity gets a lot of attention in the news, and rightly so, because when an individual gains weight, it puts them at much higher risk of a whole bunch of different disease conditions, including coronary heart disease and stroke and liver problems and high blood pressure. And all of those conspire to cause shortened lifespans and real impacts on people's quality of life as they go through life if they are overweight. And so certainly my funders are more interested in understanding human obesity than pet obesity, but why would anyone support really the study of dogs? And that comes down to the fact that actually in biology we use what we tend to call model organisms a lot. The most common model organisms in which we study biology relevant to human disease are lab mice and rats. Even fish, fruit flies, and little worms are very valuable to us understanding the fundamentals of biology and very clinically relevant things as well. And what we can do in animal models is study conditions systematically in a way that we simply wouldn't be able to in the human population. Now, compared to these lab animals, studying dogs as a model organism is somewhat less convenient, quite frankly. I have no interest in studying animals in a sort of research. I don't want to keep dogs only for research, and so all my work is done with people's pet dogs or working dogs. Those kind of free-living dogs, although it's much harder to control than the lab animals that you saw on the previous slide, actually have huge advantages when it comes to studying human disease. And that comes down to two main categories. Firstly, the similarities between dog and human diseases. And secondly, their profound differences when it comes to dog genetics. So first, those similarities. Our pets, our dogs in particular, develop many of the same diseases as their human owners, and they do it while sharing an environment with them. That means they're exposed to similar toxins, environmental factors, but also to many of the kind of more high-level environmental factors, like the increasing availability of food in our modern world. And certainly, my patients develop many of the same diseases as humans, whether that's asthma or hip arthritis that needs to be treated with a hip replacement. And sometimes they do it in a way that is particularly convenient and informative when you want to study disease not only for the dog's sake, but also for humans. So for instance, this X-ray on the bottom left of the screen shows a kind of bone cancer called osteosarcoma, which is a pretty devastating disease in dogs, and unfortunately is really quite common in large and giant breeds of dog. In contrast, in humans, it's a very uncommon kind of cancer, and so there is real potential in studying something that happens commonly in our species, in order to understand a bit more about its biology relevant to humans too. And the same goes for particular kinds of heart disease, which unfortunately are very common in some dog breeds. And those similarities are certainly true when it comes to obesity. This kind of cuddly pair, you might generously call them, two Labradors who are both really quite overweight. And unfortunately, they may not flag up as looking that unusual to you. We, as a society, have got quite used to seeing overweight dogs, and the surveys that have been done most recently suggest that as many as 2/3 of pet dogs are overweight or extremely overweight, which we tend to call obese. And that's a problem for dogs because it causes disease and shortens lifespan. That's the basic take-home message. Now, it's pretty intuitive, really, that a dog who looks like the one on the left isn't going to have as much fun and have as high quality of life as a slim, athletic beast like the one on the right-hand side. And some of that is about the ability to be athletic and express natural behaviors. But a lot of it is also about the fact that overweight dogs tend to have a higher rate of quite a lot of different diseases, either with obesity causing or exacerbating breathing and heart problems, trouble with arthritis and other orthopedic conditions. Some hormonal problems are more common in overweight dogs, and some very practical things like incontinence is worse in overweight dogs. And that's something that's quite common in my middle-aged female patients. And if they become overweight, then it can really make living with those pets really problematic. And so I study canine obesity because of its importance in my species of interest. But I study genetics in particular because of its relevance and what it can teach us about human biology. And whenever I give a talk about the genetics of obesity, it's kind of important to say up front that, well, we know the answer to this question, don't we? We know what causes obesity. If any individual, human, animal, eats more than they burn off during their daily lives, if they have energy intake that is in excess of their energy output, they will ultimately pile on the pounds. And that's basic physics. It's no surprises. There's no surprise in it, and it's something we've known for a long time. But what's interesting is when many of us have the ability to overeat, why do only some of us gain weight? And what we know is that a lot of that variability is down to our genes. And this was initially most intensively studied in human populations, whereby studying twins and families, and more recently, very big populations, we can get very accurate understanding that the tendency of each of us as individuals to gain weight and to become overweight is heavily influenced by genes. 40 to 70% of that heritability of obesity has a heritable nature. And actually, that number is remarkably consistent with about half or more of an individual's tendency to gain weight being down to genetics, whether you look at broiler chickens growing for meat, or whether you look at cattle or pigs, or, as it turns out, when I look at dogs. And so genes are important, but which genes and why? And why on earth would we have a genetic makeup that predisposes us to a debilitating problem like obesity? And the answer to that lies broadly in the fact that for the majority of evolution, dog or human, we have existed in what I often hear called a resource poor environment. You know, the dogs in this picture, the humans in this picture from a cave painting 12,000 years ago really had very little worry about becoming too fat, but really did have to make a big effort to get food by hunting, by consistently gathering small morsels throughout the day. And certainly, if we forget to eat, then we die. You know, there's no question about it. And so, consequently, the body is well set up to remind us to eat, and actually to remind us to get high value food, high energy food, with a physiological setup that tends to promote us being a bit heavier than would classically can be considered to be healthy. Because remember, in the past, periods of high food availability were commonly followed by periods of fasting. And in that context, it makes sense to have a little bit of extra energy laid down as fat to draw upon when food is scarce. And so the system that I've drawn here on a dog diagram actually could very easily be, in fact was, adapted from a human similar diagram. The systems are highly comparable between species. And what we now understand after many years of intensively studying these kind of systems in research environments is that the body monitors the energy reserves that we have in fat by sending kind of long-term signals of the body's energy storage depots in hormones, specifically leptin, but others too. And then also sends out short-term signals from the gut and the pancreas, from the digestive tract, in both nerves and hormones, which give an indication of the energy flux from minute to minute, hour to hour, day to day. And those short and long-term signals are integrated by an area of the brain called the hypothalamus, which coordinates them, and kind of the net effect of those signals is translated into the kind of feeding desire to feed, or desire to stop feeding, or the sensation of satiety that motivate our feeding behavior, and to an extent our energy expenditure as well. And of course, all of that occurs subject to environmental influences about what food is available, what it's likely to taste like, how rewarding it's likely to be. And here's the bit that I'm interested in. Every level of that biology has the potential to be tweaked by genetic mediators, which can alter one way or the other each aspect of the system. Now, as for which genes are responsible and why, well, you know, there's been a lot of effort on that over the last 20 or 25 years. There's been much insight from the study of human patients and laboratory animals who have severe early onset obesity caused by a single gene mutation. And in those cases, obesity often starts early in life and is quite severe. And studying those patients has been very informative and very important clinically to improve their health outcomes. But the take-home message from that vast body of study out there is that kind of common obesity, the obesity that is tends to kind of creep on us as we enter into adulthood, and then stick with us and get a little worse through middle age. Common obesity is broadly under the control of hundreds, possibly thousands of genetic variants across our vast genome. And where we have been able to identify which genes are responsible, they mostly affect how the brain regulates our eating behavior. and they are expressed, the proteins that the genes encode are expressed in the brain and probably modulate our perception and response to food and drive to eat. But there's still an awful lot we don't know. The largest what we call genome-wide association study done to date in about 700,000 people was vast and extremely powerfully set up statistically to discover new genes. It's found about 700 different regions across the genome. But even so, we've only explained less than 10% of the heritability of obesity. So I entered this field knowing that there was this question about which other genes are important and how they exert their effect, and proposed to answer that question by studying dogs. So you might ask why, very sensibly. I've told you that dog obesity is quite similar, but actually when it comes to genetics, it's the differences that are most important. I started out with Labradors. This is a single dog breed, a very common popular breed in the UK, and the nation's fattest dog breed as well. These guys come with a phenomenal reputation for being kind of real chow hands, very foodie dogs who will always be on the lookout for food, like that video of Woody I showed you at the beginning. And the reason I was interested in studying them is because the way that dogs have been bred since domestication from the wolf somewhere around 30,000 years ago has led to real benefits for geneticists interested in finding disease genes. So following domestication, there was a long period where dogs were largely free breeding. Yes, they lived alongside humans, and yes, humans may have encouraged them to produce different types with function in mind. So little short-legged dogs to go down rabbit holes, or big, strong, brave dogs to fight off wolves. But although they may have formed what we would recognize as breeds, these dogs were ultimately bred for health and with a job in mind. And so if there were health problems in them, they tended to be fairly quickly eliminated from the breeding population, or those negative traits not accentuated. In Victorian times, dog fancying became a fashion. And those breeds of dogs, which had developed over many thousands of years, were recognized, lauded, praised, shown, and importantly given prizes. And people desired a degree of standardization, because in that competition process, in dog showing, people wanted to know that their dog had the right characteristics, or that they could be breeding dogs with the right characteristics to bring home the rosette or the trophy from the next dog show. And in the process of formalizing what were called breed standards and formalizing dog breeding in recording it in a stud book, the Victorians unwittingly caused what we refer to as geneticists as population bottlenecks. And so that really rather a few founder individuals were forced through a narrow funnel into and became the founder populations of modern dog breeds. And that's had some really quite important consequences for the future. So we now have these extraordinary and really quite extreme diversity of dogs with different sizes and shapes, different personalities, different fur types, all of which are very similar within one breed, but very different and diverse across the breed. And from a geneticist point of view, the existence of old bottlenecks many thousands of years ago at the domestication from the wolf, and these recent bottlenecks at the point of breed formation in the Victorian era, has led both to problematic ill health, which isn't something I've got time to dwell on particularly in this talk, although it is important to say that the high food motivation and obesity prone nature of Labradors is part and parcel of that. not only do we see those ill health things, but actually it's got somewhat of a flip side of that, is that there are benefits, because as geneticists, we are able to track down disease-causing genes rather more straightforwardly in dogs than you can in a very diverse human population. And that comes down to the fact that when we go looking for the gene or the mutation causing a disease, essentially what we do as geneticists is compare a bunch of dogs who don't have a condition with a bunch of dogs who do have the condition. And we try and look for something which is common to all the affected dogs, which is absent from all or most of the unaffected dogs. And if we do that in a very diverse population, like a bunch of humans from the UK or across the whole of the dog species, then it's really difficult to pull apart those patterns. But if we look within a modern dog breed, they tend to actually all be so similar that it's pretty easy to pick out the disease-causing mutation, which I've represented by these orange dots. Now, you may be able to do that in a more diverse species because those disease-causing mutations are likely still to be there, but it's an awful lot harder to pick the pattern out. And so you can see that there is a good reason to study genetics of a trait within a breed. And so that's what I did in the Labradors. And actually, I did something very simple in the first instance with the Labradors because we know quite a lot about obesity biology. So I went off and I sequenced some genes that I thought might be good candidates for causing the obesity phenotype. And what I found when I contrasted the genetic makeup of a few lean dogs with a few fat dogs was that there was a mutation in a gene called POMC, which was pretty uncommon in the thin dogs and really quite common in the overweight ones. And that mutation is a deletion in a gene. We can be very confident that it would disrupt this gene POMC. And the reason I'd gone looking there in the first place is because it's part of this, which is a brain signaling pathway, which is quite central to regulating the body's food intake, energy expenditure, and ultimately body weight. And so we call this the leptin melanocortin signaling pathway. And this little cartoon over here is meant to be fat cells in the body. And fat cells produce this hormone leptin in greater amounts when there's more leptin around. And so leptin is released into the bloodstream and acts in the hypothalamus in the brain on nerve cells which produce this protein POMC. And when those POMC neurons are activated, they release downstream products of POMC. I've got a diagram of this later, called melanocyte stimulating hormones, with the net effect that when there's masses of fat, masses of leptin, then we see signaling through the pathway and a reduction in food intake and an increase in energy expenditure. Now, actually, what we know is that this pathway is most active and most important when it's disrupted. When there isn't very much leptin because there isn't very much fat, the body sends out a starvation signal from this absence of leptin. And when there is disruption of signaling through this pathway, there is a massive increase in appetite and, to an extent, a decrease in energy expenditure, although that hasn't been very well proven in humans. It had been suggested in mice. And what we found in the Labradors is that there is a mutation in this gene POMC, and it disrupts signaling by melanocyte stimulating hormone at MC4R, melanocortin 4 receptors. And therefore, we would hypothesize that this mutation I found would cause precisely the kind of foodiness, food obsession that we see in the Labradors, and possibly that it might affect energy expenditure too. And so we went out to test that. Initially, I discovered this in just a handful of dogs. It was important to go and look in a bigger group. So in about 250 dogs, we looked at the effect of carrying 1 copy or 2 copies of this mutation in the gene POMC. And what we found is that in dogs who had 2 copies, they were on average about 4 kilograms heavier. Bear in mind your average Labrador is somewhere around the kind of 25 to 30 kilogram mark when they're at a healthy weight. So that's a pretty big effect. And we can express that not just as weight, which of course could be to do with how muscly or how tall a dog is, but also using something called a body condition score, which is a measure of adiposity, of fatness. And so the difference between dogs with no copies and 2 copies, all else being equal, is about a body condition score point, or about 10% extra body fat mass. Of course, we studied these guys because of their legendary foodiness. And so I spent quite a lot of time developing a questionnaire which put a kind of number on greed. And sure enough, and gratifyingly, owners reported that dogs carrying this mutation were more highly food motivated in the home environment. And so we published that work some years ago, but actually in the intervening years, I was quite interested in trying to drill down more specifically into the effect of this mutation on the neural pathways controlling appetite. And we've done that by trying to dissect out different aspects of feeding behavior. So first, we looked at how much dogs would eat when given free access to eating essentially as much as they liked. So we gave them a series of meals, of cans of dog food, and saw how much they ate. And although all dogs in our studies, whether they had no copies or 1 copy of this mutation, ate an enormous amount, in the order of 2 kilograms of food at a sitting, actually there was no statistically significant difference between the 2 groups. Those dogs with this mutation did tend to eat more, but we couldn't, it's not a huge difference. Where we did see something more interesting was when we used a different kind of test to try and get a handle on how hungry these dogs became. And so what you can see here is stills from an experiment where we looked at what we call motivational salience in response to a food cue. And so what you can see in this video is my colleague Gabby showing a hot dog sausage to Boody, who is a dog who is what we call wild type for this mutation. So she doesn't have a copy of the mutation. And you can see that she was pretty interested in the sausage, and she kind of pays attention to the box, but actually fairly quickly figured out that she couldn't get to the sausage and spent her time exploring this new environment. And that was reasonably typical of the dogs who came and took part in our study who didn't have a copy of the POMC mutation. Contrast that, if you will, with Ash here, who does have a copy of our POMC mutation. And Ash spent the entire of the 5 minutes of the video capture study really very excited and somewhat kind of frenetic in his pursuit of the sausage. I've turned the audio off this video because you kind of hear him squeaking away in the background. Now, intuitively, you can see that there is a difference in the behavior of those 2 dogs. And of course, we chose kind of good examples to show you for this kind of presentation. But importantly, these data have all been coded painstakingly, actually, frame by frame, to put a number on the kind of interaction with the cage versus with the little box versus exploring or kind of sitting down near owners or whatever. And what we can do when we graph that out and test it statistically is show that there is a clear difference in the way the dogs with the mutation respond to the sausage compared to those without the mutation. And because we did this in a standardized environment at a standardized time, about 3 hours after they'd had a meal, then we can say that the one big difference between those 2 groups of dogs is their genotype, and that we are seeing this difference because these dogs are more highly motivated by the food item, which is most likely because they are hungrier. Now, what about the energy expenditure? Now, I must admit, when I started doing this experiment, I really didn't expect to find anything. When people have looked at humans with sort of similar mutations, they've not, it's not been looked at in great depth, but there weren't striking differences in energy expenditure. So you'll notice that the dogs in these pictures are not Labradors, but flat-coated retrievers, because they're the only other breed that we found have this mutation. And as it happens, it's more common to find dogs with 2 copies of the mutation in the flat-coat population. And so we recruited some of these lovely dogs and got them used at home to hanging out in our respiration chamber, which, as you can see, is a sort of modified air transport carrier actually for dogs. And what you can see in that bit of kit in the middle is a sealed chamber with very controlled airflow coming in at the bottom, at the front, and out at the back, that is pulling air through at a known rate and into a gas analyzer, which allows us to measure the amount of carbon dioxide that these dogs breathe out, and in turn to therefore calculate how much energy they use when they're at rest. So we wait until the dogs are fast asleep in the chamber and then measure their energy expenditure. And when we did that, we had a very striking finding. So I'm afraid I couldn't think of a really good way to show you this without facing up to a graph. But what we have on here is a graph that shows body weight on the x-axis horizontally at the bottom. And it kind of figures that we have that in there, because if you're a heavier dog, you're going to use more energy just keeping your cells alive than if you've got fewer cells because you're a lighter dog. And that's why all of this, because it's metabolic rate on the y-axis going vertically, all these points tend to sort of slope up and right. But what we can see on here is that wild type dogs and those without the mutation, who are shown in white, tend to sit much higher up on the graph than those affected dogs with 2 copies of the mutation, shown in black. And actually that difference is really striking. It says that when these dogs are lying in a cage at rest, doing absolutely nothing, the dogs with the mutation are burning off about 20% less energy than their wild type counterparts. That's a big difference. I've tried very hard to make these data go away because I didn't quite believe them to begin with, but I can't. It's real. And actually, it fits very neatly with what we know about this leptin-melanocortin signaling pathway. If it's broken, then the body is essentially receiving a physiological starvation signal, its response to which is eat as much as possible and conserve energy. And that's what we're seeing in this Labrador and flat-coated retriever mutation, which we have shown is a major modifier of weight and fatness. It affects both hunger and metabolic rate, and it's doing so in a fairly striking way. Now, it's important to say that actually only about 25% of pet Labradors carry this mutation, and yet people commonly say, it's not true, but they commonly say, oh, all Labradors are really foodie. And so there's plenty of other genetic stuff going on in Labradors, and I'm working on trying to find what they are, doing my best. But I think it's a good moment to reflect on those responses and what we can learn from Labradors that's relevant to our veterinary care of these patients, and also just to our understanding of obesity in general. And actually, this quote here is taken from a paper that reported the clinical features of human patients with POMC mutations. And here it said both patients were markedly hyperphagic, they ate a lot, as indicated by a score of 9 on a Likert hunger scale ranging from 0, no hunger, to 10, extreme hunger. So they felt hungry all the time. Psychological evaluations revealed this led to constant extreme dissatisfaction with their quality of life. Being hungry all the time is blimmin' horrible. And actually, I think this does present us with some quite real challenges in managing dogs with these mutations. Because when I was at vet school, we're taught about the 5 freedoms of animal welfare, 2 of which are freedom from hunger and freedom from disease, and obesity is certainly a disease. And so there's a real tension here between restricting food to keep dogs at a healthy weight and making them feel hungry all the time. And I think there are some very practical management interventions we can apply in affected dogs that I'm happy to answer questions on at the end, but which I think will improve clinical outcomes and welfare in these patients of mine. And from a practical point of view, it certainly means that owners of highly food motivated dogs, certainly carrying this mutation, are up against it when it comes to keeping their dogs slim. These dogs are opportunists. They are on the lookout for snacks at every possible opportunity, and they will work really quite hard, whether it's going off to find extra bits of food or just sitting there gazing adoringly and giving their owners the big brown eye treatment until they get a snack. And they really make an effort to get food, and it makes it much harder if you own a really foodie Labrador to keep it slim than it would be if you had a much fussier breed of dog. So all of that's kind of clinically important and relevant, but it wasn't what made me a magazine cover girl for the only time in my life. And the reason why this kind of high impact journal was interested was because of some quirks of POMC biology, which are particularly helpful when it comes to studying dogs as an animal model of human disease. So I mentioned earlier that POMC is quite a complicated gene in that it makes a protein, which I've indicated by the blue bar at the top here, which initially is a long molecule, but is cleaved, cut up by a number of enzymatic reactions into a whole bunch of different brain active fragments over a fairly complex processing effort. And the headline acts when it comes to obesity regulation or energy metabolism regulation in humans are these two, alpha and beta MSH. And they act at those melanocortin 4 receptors that were on our diagram. And we know that humans who lack either alpha or beta MSH are prone to overeating and becoming very overweight early in life. But actually, the number of human beings who have been identified with beta MSH mutations is very small. And certainly there haven't been enough beta MSH mutation human beings to study systematically. So trying to work out what the difference between alpha and beta MSH and how each of them, these two very similar molecules, each are necessary and do something perhaps different when it comes to managing energy metabolism in the body. Studying that systematically has been quite difficult and impossible in humans. It's also been impossible in rodents, because through a quirk of evolution, rodents don't make beta MSH. They lack one of those cleavage sites, and rodents get on just fine with only alpha MSH and never produce beta MSH. Enter the Labrador, where in normal, and in all dogs for that matter, in normal dogs, they all produce both alpha and beta MSH. But in a quarter of pet Labradors and 2/3 of flat-coated retrievers, there is a mutation which, because of its nature, scrambles the whole of the end of this protein that's produced, and means that we can confidently say that while they have normal alpha MSH, they have no beta MSH. And so the smoking gun when it comes to causing Labrador obesity is this absence of beta MSH, and it gives us the chance to try and understand through those studies I've described how it is that beta MSH has its most important activity in the body. And so the Labrador example is quite extreme, really. It tells us about a really big mutation that kind of is very disruptive to the protein which the gene encodes. But it's worth pointing out that actually there's lots and lots of examples of less extreme, but equally clinically relevant disruptive genetic variants, which are relevant in humans too. And so all of the things in this table talk about genes that we kind of know what they do. So CCK, for instance, is a satiety hormone. So it's produced after a meal in response to protein in the gut, and signals to the brain that it's okay, we've got nutrition, and now you can have a sensation of satiety, fullness. And there are common variations in the gene CCK out there in the human population that, if you carry, are associated with extreme meal size. So you don't feel full until later. Similarly, there are variants in the leptin gene associated with extreme snacking behaviour. I don't think that means eating your chocolate bar off the edge of a cliff or something. It's more about that sensation that actually I feel very familiar with. That I always have an eye on my next meal. I always know that it would be quite nice to have some food next. And I'm conscious and mindful of that, but I'm aware that it's actually quite different to my husband's sensation of food, and where he will eat well and likes to eat big meals, but he doesn't plan the next meal. He's not that bothered about snacking in between. And so these littler, more minor variants in genes are out there in the population, and each can have an incremental effect on our individual response to food, and therefore our tendency to overeat or not. And I think it's important to recognize that these are there, and that these altered or varied sensations and responses to food are real, they're hardwired, they're genetically driven, and it's nothing to do with people's moral compass or willpower. It means perhaps that those of us who are always thinking about food in between meals need to use a bit more willpower to overcome that and not to snack all the time. But even if we can do that most of the time, if we're up against a bigger drive, we're likely to slip more frequently. And I'm jolly well blaming a suspected leptin variant for my lockdown extra tire or two around my waist. And overall, what this does is point to that take-home message that genetically driven, hardwired differences in our biology and our responses to food underlie our neurological response to food, and therefore our behaviour, our eating behaviour and our food choices. And even if those changes are very subtle from one person to another, if they are in play all day, every day, for year after year, it will ultimately mean that some individuals are prone to sneakily gaining weight as time goes by. The other thing I think we can kind of quite objectively believe from studying the dogs, in a way that perhaps people are somewhat resistant to taking on board when it comes to thinking about human obesity and human biology, is this effect of the environment in which we find ourselves in terms of making food choices. And it's worth going back to that genetics and evolution story to think about the pre-domesticated dog, wolves, and how hard they have to work to get a decent, high-calorie, nutritious meal. Or even to think about the early domesticated dogs. This is a picture of a village dog in India who's probably living as domesticated dogs have for in the order of 30,000 years, albeit with plastic bags in the picture. But, you know, pottering around a human settlement, trying to find discarded scraps of food that are worth eating. Probably not going to get great tasty things there. And it makes sense to have a pleasure response in response to it, when they get high calorie, high energy food, that will drive them to keep working hard to find those little morsels. And if you take a body that is evolved in that background and put it into a modern pet dog's world, where there is a plethora of food marketed towards pet owners, most of which talks about how delicious it is and how much your dog will like it, then it's hardly likely that they don't turn down whatever they're offered in their bowl. And then if all the hard work they have to do these days to get a little bit more food is to sit there and gaze adoringly at their owner, then, you know, it's hardly any surprise that they go to that effort. And to put that back in the Labrador retriever story, I mentioned earlier that the only other breed we found this in was flat-coated retrievers, and we can tell from the genetics that it was likely a variant that was inherited from a common ancestor, and it seems likely that that common ancestor was the St. John's water dog. And these were fishermen's dogs who went out on the boats in 16th century or 17th century Newfoundland, leapt into the icy waters, pulled in the fishing nets, and worked incredibly hard for a living. And you'd have thought that in that context, a willingness to kind of snaffle any rotten fish head that was coming along would probably do you no harm. And certainly a layer of blubber, if you managed to develop one under your skin, would perhaps keep you warm as a dog rather than causing any trouble. We've also got a hint from the data in the Labradors that because this is more common in guide dogs, assistance dogs, maybe if you're a bit foodier, you're a bit more willing to work for humans. And that may have played in to Labrador's appeal as gun dogs down the centuries. And it's really only in the kind of modern day that we see Labradors affected by a debilitating obesity as a consequence of being so highly food motivated. So their environment has changed, not their genetics. And again, if we kind of turn our understanding of that hardwired biology in dogs to the human context, I think very few of us would pile on the pounds if all we had to eat was vegetable soup. We might be perfectly healthy. We probably would be very healthy, and it looks delicious. But I'm pretty confident that none of us would massively overeat to the point of obesity if presented with nothing but vegetable soup. Whereas if we were presented with cake, or as we are regularly offered sort of tempting high calorie, high sugar, high fat foods, then it's much easier to take on excess calories. And actually we get a real sense of reward and good feeling from eating something quite that delicious. And so that's a sort of binary choice of what you're offered. But realistically, we are offered delicious things every time we go to the supermarket. And most of us in the West, at least, are offered, have the ability to eat large volumes of energy-dense food whenever we want to. And it's no surprise, perhaps, that we therefore tend to make choices that promote obesity across the population. So just pulling all of that together, I'm interested in studying the biology of obesity, both to help humans and to help my veterinary patients as well. And we know that obesity causes a huge number of health problems in both species, and that genetics are very important in determining whether one individual is susceptible to becoming overweight, whereas others seem to be quite relatively resistant to developing obesity in this modern environment that we live in. I talked about the fact that dogs can be really good models for studying human disease because of their similarities in terms of the diseases they get and the environment in which they get them, but also because of their genetic differences and the way that dogs have been selectively bred down the centuries means that there are those genetic quirks that makes gene mapping quite tractable in my patients. We've studied that in the context of the retriever and his tendency to gain weight, and shown that a mutation in POMC has not only promotes food intake by making these dogs much more hungry between meals and more highly food motivated in their home, but also remarkably by having this big effect on energy expenditure too, which is I would stress unusual. You know, the take home message from this really is that in general genes affect our perception of food and the sensation of what it feels like to eat when we feel full and so on. And those differences between individuals' perception of food and how it makes us feel are real. and biologically hardwired. And given that we evolved in a resource-poor environment and now find ourselves faced with plentiful, high-calorie food wherever we go, then it's perhaps no surprise that there's such a problem with obesity in both humans and dogs. Now, with any research talk, there's always thank yous to give. And the first and most important one in this talk is to say thank you to all the owners who've contributed to our study. We've gathered in the order of 2,000 dogs' DNA samples in the pursuit of this science, and people have been absolutely wonderful at contributing information and slobber from their dogs in order to allow us to do these studies. And then, of course, some owners have been particularly dedicated and amazing to volunteer to get their dogs used to the energy expenditure chamber, or to get them eating the right diet so that they can come and take part in our food experiments. And a lot of that work was done by Jodie, Marie and Gabby, who you can see in those pictures, who have been the researchers who've gathered a lot of the data as it went along. So I'm very happy to answer any questions now. You can find us on social media if you want to hear any more, and certainly do get in touch if you watch this on a recording and are interested in participating or finding out more.
Thank you very much for a really great talk. As you mentioned, we'll be taking questions. So if you do have any, please put them into the chat and I can read them out. For which you only have one. So Athea Maybury asks, why are both your obese figures female? Just asking. Great talk.
I don't think they are. Certainly the male-female figures I've got here are, I think, supposed to be a male and female differently. So it's important. And actually there are sex differences in the health consequences of being overweight in men and women, and there are differences in the nature of obesity and risk factors. And from a genetics point of view, it's been quite late in the day that people have kind of considered those and considered different populations separately. So it's something that we're all very aware of in the field.
So I was really interested to hear, especially the ancient origins and the bottleneck effect of the genetics. And so one thing you mentioned is that hunger reflex was likely developed and likely encouraged genetically in the past. So do you think maybe in 10,000 years, we may have the opposite problem, especially those who perhaps stay in a resource abundant environment for that long?
Yeah, it'd be fascinating to think, wouldn't it? I mean, certainly when I was first introduced to this field, the idea of a thrifty gene hypothesis, that our ancestors did evolve in a resource-poor environment and had very little selection pressure against becoming overweight, but did have a selection pressure against not eating enough, is a very compelling hypothesis. I think it's important to acknowledge that actually there are alternative genetic hypotheses that we may have ended up like this through a process called genetic drift, whereby stuff just kind of comes up in populations, and so not all of the obesity proneness that it comes through via genetics is necessarily down to that ancestral selection pressure, but it seems quite likely. Yes, if we hang around long enough, then I think there is likely to be a pressure, and particularly because, as well as the kind of well-publicized risks of diabetes and heart disease and stroke, which we tend to think of as middle-aged to older age conditions after reproducing, there are also impacts on fertility from being overweight through polycystic ovary syndrome and other mechanisms. And so, yes, there is potentially a fertility impact and therefore a selection impact from being overweight.
Interesting. One other thing, with the accessibility of genetic tests now getting easier and easier nowadays, do you think there'll be specific testing and perhaps treatment for obesity on a genetic level with patients?
Yes, so that's real and here with us today on several levels. And so my colleagues at the Institute of Metabolic Science, particularly Stephen O'Reilly and Sadaf Faruqi, have for many years studied patients with severe early onset forms of obesity and tried to determine what genes are responsible and how they exert their effect. And that's been hugely informative to the field in understanding this biology, but it's also been very important to those individuals who often have been unwell or sort of debilitated by their obesity from very early in life. And because of the nature of society being very judgmental about overweight people, have often had a fairly miserable kind of emotional consequence to that as well. And by providing those patients with a genetic diagnosis, it can be a hugely valuable boost. In addition, in some cases, they've also been able to move on and provide tailored specific treatment as a consequence. And so, for instance, we learned a lot about the biology of leptin from the study of patients who have an inability to produce leptin. And in those patients, Steve and Sadaf were amongst the first to treat with external, chemical produced in the lab leptin. You can be injected and can reverse that early onset obesity and a plethora of other problems that come alongside it. So that's a huge and important thing. And actually recently it's been in the news about a drug called setmelanotide, which has been developed that acts on melanocortin-4 receptors. And so patients with mutations in POMC or patients with melanocortin-4 receptor mutations as well, and various other things have benefited from this very recent development in pharmacology that can treat their severe obesity. So drug testing has that very kind of precise effect there, but also on a more general basis, you can now, many of us, lots of people have had 23andMe or other similar sets of genome-wide testing, and those direct-to-consumer genetic tests are out there. And if you take them, just as they may tell you if you have particular cancer susceptibility genes, they will also tell you if you are particularly likely to be prone to obesity. And I think there was a bit of a worry in the field that if you told people they were obesity-prone because of their genes, then they'd just kind of give up, roll over, and say, well, I'll eat what I like then. Whereas actually, I think there is good evidence out there that it tends to either empower people to work harder to combat weight gain, or at least has a neutral effect, just allowing them to understand their tendency to overeat.
Yeah, and I think that really raises an interesting question for this field and many others. For, of course, if you have particular obesity or some other condition, you test for it. But what about for those who don't have any symptoms? Do you test for all these things all the time? Do you have any thoughts on that?
So would you do a genetic test for something that you don't have already?
Mm-hmm.
I mean, I don't know that there'd be much clinical reason to do that, would there? But certainly things like 23andMe allow people to get testing for something that they don't have already. That has ethical implications because you tell someone that they might get a problem in the future. With obesity, I think that's less of a problem because people aren't quite so afraid of it, and they understand that ultimately we have control on what goes in our mouths, although it can be a lot greater effort for some people to exert that control than others. That's a bigger deal with cancer testing, for instance. Should you, if somebody, the kind of classic thing is that someone gets bought a genetic profile as a kind of quirky birthday present and then discovers that they are prone to be a particular kind of cancer, and that can be quite a frightening experience, but perhaps less relevant with obesity. It's been something that we've thought about hard in the dogs as well, because having identified this in the Labradors, we know that it's real, that it has an effect to have this mutation, but that ultimately lots of owners of dogs carrying the mutation can control their dog's exercise and food and keep them at a brilliant healthy weight with a good quality of life. And so should you test for that? Would it make people work harder to keep their dogs slim, or might it just mean that they give up? And we have dealt with that by offering the test, but giving really quite intensive support literature that goes out with the results, so that people can have support and confidence that they know how to control their dog's weight if they carry this